So, what we're going to do is review some of the most frequently missed questions historically in the Reproduction and Inheritance unit exam. So, I'm really going to ask that you click slowly through this slide presentation so that you can kind of quiz yourself as you go. So, remember, in single celled organisms like amoeba, paramecium, bacterium, ask yourself, why do those organisms go through cell division? And hopefully you would be able to say for reproduction so that they can make more amoebas, paramecium, or bacteria. So this diagram shows one cell going through cell division and through the series of steps eventually splitting into two separate cells. So one cell in diagram 1 reproducing and splitting into two cells, diagram 6. So the concept of twins, both fraternal and identical, can throw people off a little bit, and you're really going to want to make sure that you study the diagram so that you can really understand what makes fraternal twins and what makes identical twins. Remember, identical means exactly the same. So if you have two eggs and two totally different sperm developing at the same time, then clearly they don't have identical DNA, and those would be considered fraternal twins, not identical, but like siblings. So they're no more genetically alike than any other siblings born at separate times. Okay. Identical twins, again, have identical or exactly the same DNA. And the only way that can happen is if there is one sperm and one egg. So there you have your zygote splitting kind of an extra time to get you identical copies or identical twins. So again, if you study this diagram, you can see the fraternal twins, two separate eggs released from the mother at the same time, or the female, two separate sperm. So it is not genetically identical. Whereas an identical twin is kind of like nature's version of clones, one egg, one sperm. The zygote splits an extra time. You have two separate yet identical developing organisms. So one of the other concepts on the unit exam that students struggle with a lot is cloning. And this diagram is showing you a cloning procedure where the baby rat will have the traits of question mark. So go ahead and pause for a second and really study this diagram. Pay attention to the steps that are there. So push pause and really identify in this cloning procedure, the baby rat, okay, is going to have the traits of which adult rat? One, two, or three. So hopefully you were able to look at the diagram and notice that the brown rat, rat number 1, the nucleus of its egg cell was removed from rat 1. So hopefully you thought to yourself, okay, the nucleus is where the genetic material is found. So if the nucleus of egg 1 was removed, then so is its genetic material. The gray rat, rat number 2, is its skin cell was taken, and the nucleus of the skin cell was removed. So again, with the nucleus goes the genetic or DNA, the genetic information or the DNA. So then what it looks like it happens here in step 3 is that the nucleus from the skin cell, which has the DNA of rat number 2, was then put into the egg cell of rat number 1. Remember, that egg cell, that DNA was gone. The nucleus from that egg cell is gone. So the nucleus from the skin cell was placed into the enucleated or empty egg cell. Step 4, that egg cell with the genetic information from rat 2 was then placed into the womb of rat 3. So all rat 3 did was provide a nice, safe environment for the baby rat to develop. It did not provide any genetic information. So in step 5, a new baby gray rat was born. So hopefully you were able to tell us rat number 2, the gray rat. Okay, looking at the development of the corgi. how it developed over time. This is the process when humans select certain dogs or organisms to breed with each other. And hopefully you're able to identify that as selective breeding. So one of the things that we're really going to talk about more next unit, you will hear the term tissues on the unit exam. So I just wanted to make sure that you kind of understood where the term tissue was coming from and kind of put it into context if you do hear it on the unit exam. Okay, so all cells are made up of organelles. Those are your cell parts, your nucleus, your mitochondria, your vacuoles, okay? A bunch of cells make up tissue. So your heart cells make up your heart tissue. Your skin cells make up your skin tissue. Tissues then make up your organs. Organs make up the organ system, like all the organs of the digestive system make up your digestive system. And then all of those systems together make up the organism. So I really just wanted you to be able to understand that cells make up tissues. So just kind of put it in context if you hear the word tissue on the unit exam. So yes, cells are made of organelles. Yes, no, tissues are not made of organs. Organs are made up of tissues. And are tissues made up of cells? Yes. And again, that's the big takeaway that I want you to get from this. All right, looking some examples of cell division. This first diagram is showing a plant cell undergoing cell division or mitosis. This is an animal cell going through cell division. So the question is, what can you say about this diagram? So if you were to study this diagram and looked at a plant cell and looked at an animal cell, hopefully you would be able to come to the conclusion that plants and animal cells undergo a similar process, cell division. All right, so let's just take a look at another Punnett square example. Just again, covering kind of those most missed concepts. So in mice, gray fur is dominant, white fur is recessive. So if two hybrid, that means two different, a mix of genes, capital G, lowercase g, mice mate, what percentage of their offspring will be white? So hopefully you can pause this and go ahead and complete this Punnett square. So hopefully you were able to say that you were going to cross two hybrids, putting the parents on the outside of the Punnett square. So see again, this parent here, this parent here, and then you go ahead and you complete the Punnett square. And you can see that inside the Punnett square represents the possible genotypes of the offspring. And in this particular case, when you have two hybrids that are mating, you could, it's totally up to chance, but you could get a pure dominant, you could get a hybrid, or you could get a pure recessive. Again, completely up to chance, but you can identify percentages. If gray is dominant and you're looking for white, the recessive trait, it would have to be little g, and that would be 25% chance. All right, pedigree charts. You are going to be expected to interpret pedigree charts on the unit exam. So I'm asking you to study this pedigree chart and tell me what is wrong with it, okay? And it's in the parents' generation. So go ahead and push pause and study this diagram, this pedigree chart, and see if you can determine in the parents' generation. Remember, this is the first generation. Down here is the second generation. Go ahead and push pause and see if you can determine what's wrong with the pedigree chart. So hopefully you were able to start assigning some genotypes to this pedigree chart. And you would have to note that a pure recessive offspring had to get one recessive gene from mom and one recessive gene from dad. And clearly, dad is pure dominant.